We've all heard about the blood-brain barrier, but is there a blood-bone marrow barrier? My guest, Professor Tsvee Lapdiot, has been studying stem cells, normal and leukemic ones, for the last 20 years and joins me now. Thank you so much for joining us.
It's my pleasure.
So how did you fall in love with stem cell research?
Yes, as you know, we are all born without spare parts, and in order to cure and for clinical reasons, we want to make a difference. And stem cells have the power to do so. They are like the Peter Pan of the immune and the blood systems because they have eternal youth. And blood-forming stem cells are clinically transplanted for over 70 years, with over 1 million people being transplanted. And so this is what attracted me to hematopoietic stem cells. And of course, unfortunately, there's also a dark side to stem cell research, and this is the malignant stem cells, the cancer-initiating stem cells, which we would like to eradicate in order to cure patients.
And it'd be lovely for our viewers if you could just remind us of your work with John Dick in Toronto.
Yes. So blood-forming stem cells are characterized and identified based on their function to initiate the entire immune system and to generate all mature blood cells in order to replenish the blood circulation. And so during my postdoc training with John Dick in Toronto in the 1990s, we developed functional preclinical models where we transplanted immune-deficient mice, first with normal human hematopoietic stem cells, and later on with human leukemia-initiating stem cells in order to learn about their features, to characterize them, and to see similarities and differences between these two distinct stem cell populations in order to try and selectively eradicate the malignant clone while maintaining normal blood-forming stem cell function.
And can you give us some examples of the aspects that you study and a bit more about that model?
So in this model, the stem cells are injected into the veins of the immune-deficient mice, just like in clinical transplantation, where the mice are first preconditioned with total body sublethal irradiation. And very interestingly, the stem cells which are injected into the circulation, they find their way home to the bone marrow on their own. And this is the major reason clinically why this is the only stem cell that has been in the clinic for over 70 years, because these cells are independent in their navigation. They have a homing navigation potential, and then they can rejuvenate the transplanted recipients and regenerate the entire immune system and the entire new blood cells throughout life.
And in terms of clinical relevance today, how is that translated into clinical practice?
There are many clinical aspects to this research. In the clinic, cells are recruited, stem cells are recruited from the bone marrow to the circulation. This is a process which is called mobilization, and you can do this in this transplanted immune-deficient mice with human stem cells and learn the mechanism. You can also learn the mechanism for the homing process in order to improve it, in particular in cases where there are not enough stem cells for adult patient transplantation, for example, when umbilical cord blood for newborn babies are used. And of course, one would like to learn the features of the leukemic-initiating stem cells, and these leukemia-initiating stem cells are chemotherapy and radiotherapy resistance, and a better understanding of these cells and their interactions with the bone marrow microenvironment will help us to develop new clinical protocols in order to better eradicate them. And already we know that the aggressiveness of the human disease in these immune-deficient transplanted mice correlates with the prognosis and clinical outcome of the patients. So we can test new drugs and new approaches first in these models before we apply them clinically.
That's great. And now moving on to the actual blood-bone barrier, could you tell us a bit about your recent discoveries?
Yes. So we know for many years that there is a blood-bone marrow barrier. For example, mature red blood cells are only in the circulation, and also platelets, which are needed for clotting, are also only in the circulation. However, if you irradiate a patient for clinical transplantation, or you irradiate a mouse for preclinical experimental transplantation, In both cases, the barrier is transiently destroyed, and mature red cells and platelets can penetrate into the bone marrow. However, this is transient because the barrier would later recover. And we were interested in the architecture of the blood-bone marrow barrier and the different features, because in the river of blood, there are many mature red blood cells and platelets and other cell types, and they can secrete pro-inflammatory factors which will increase the metabolism, in particularly reactive oxygen species. And the stem cells are usually maintained in a non-motile, non-cycling, quiescent, low metabolic mode, so they need to be protected from the river of red blood cells and platelets. And this is done by less permeable blood vessels in the bone marrow that protect the stem cells and provide them with a niche. On the other hand, we need proliferation, differentiation, and replenishment of new mature blood cells continuously, and this is done mostly in more permeable blood vessels in sinusoids, where there is more permeability to the blood, and metabolic signals can penetrate and recruit stem cells to proliferate, differentiate, and egress into the circulation. And interestingly, near some of these sinusoids, there are giant cells called megakaryocytes, which produce platelets, and they also control and regulate stem cells by secreting a wide variety of factors which maintain the blood-forming stem cells undifferentiated. So near megakaryocytes in sinusoids, we can also find quiescent, non-motile, long-term repopulating hematopoietic stem cells.
Wow, and again, what could this potentially mean for the clinic?
So a better understanding of the barrier, where the entries and exits are, will better help us to develop new clinical protocols in order to improve the efficiency of stem cell homing and repopulation. Because currently, all the transplanted patients worldwide have only about 10% blood-forming stem cells compared to healthy individuals. And anything that can further increase this 10% levels might have a critical clinical outcome and improve the cure rates.
And your work looking at the secrets of normal and leukemic cells, you talk about light and dark cues, which sounds very exciting. Can you tell us a bit about that?
Yes, light and darkness cues are very important. We know that we need to sleep and rest before we wake up and work, and the same is true for our immune system. For example, there was an important experiment which was done in the early 60s where they infected mice with bacteria, either during the day or during the night. And during the night, almost all the mice survived, while during the day, almost all the mice died. And you have to know that mice are nocturnal, they are active at night, so their immune system is better active at night. So again, during the active immune system period, they are better protected. And another good example is our bone turnover. As you know, our bones are continuously degraded and rebuilt, and immune cells of the myeloid lineage, monocytes and macrophages, they fuse together to form these giant cells which are called osteoclasts. And osteoclasts degrade the bones, while bone-lining osteoblasts build the bone. And bone turnover was also shown many years ago to be regulated by light and darkness cues. Our bones grow during the rest period at night, during the darkness, while our bones are distracted during the light time. So a better understanding of light and darkness is important for bone turnover, for bone growth, and for host immunity. As you know, during aging, in particular women after menopause, they become osteoporotic. Their bone volume becomes thinner, and this is accompanied by anemia and reduced host immunity. And many old people die of opportunistic infectious disease during wintertime. So better understanding of the light and darkness cues might improve our quality of life, because we need the light for vitamin D, which is important both for our bones and for our immune system. However, we also need the dark signals headed by the dark hormone melatonin and others, which are needed for our recovery and replenishment of the reservoir of undifferentiated stem and progenitor cells, both for building new blood and immune cells, as well as for building new bone cells.
And presumably that has some clinical relevance in terms of timing of the stem cells and the treatment. And is that what you're saying in terms of day and night?
Yes,
Yeah.
this is very correct. There are reports by the group of Simon Mendes-Ferrer and Paul Frenet showing that in the mouse, the sunlight induces the nervous system to secrete norepinephrine, and norepinephrine then leads to a cascade of stem cell migration, proliferation, differentiation, and development, replenishing the blood with new mature cells on a daily basis in mice peaking at 11 o'clock a.m. in the morning.
That is absolutely fascinating. Well, listen, I'm blown away by the kind of model that you've created, and thank you so much for joining us, Professor Tsvee Lapdiot. Thank you very much indeed.
Thank you, Ian,
Thank
very
you.
much. It's my pleasure. Thank you.